JCE Advanced Level Chemistry Practical Series Experiment number 42. Extraction of essential oils by steam distillation. In this experiment, we will be extracting cinnamon oil from cinnamon leaves by steam distillation. And the aim of this experiment is to develop the skills required for extraction of essential oils from selected natural resources. Let's move on to identifying the equipment and the chemicals we will be using for this experiment. Let's identify the raw material and the equipment we will be using for this experiment. We have here cinnamon leaves. Cinnamon leaves will be used for this extraction, and when you carry out this essential oil extraction from cinnamon leaves, the essential oil that is extracted is mainly going to contain an oil known as eugenol. So in this particular experiment, we will be using cinnamon leaves to extract cinnamon leaf oil. These are raw cinnamon leaves, and we have here cinnamon leaves that are dried. We will be using dried cinnamon leaves to make sure that we can extract the most amount of cinnamon leaf oil with the use of dried cinnamon leaves. So in this experiment, we will carry out the essential oil extraction process with the use of dried cinnamon leaves. Apart from that, we will be using measuring cylinder, a beaker to measure the cut pieces of cinnamon leaves, or to measure the mass of the cinnamon leaves. We'll be using a petri dish. When carrying out the distilling process, we will be using a distilling flask. We will also be using a round bottom flask for distillation as well. We will be using a separatory funnel, or also known as a separation funnel. We'll be using a three-way adapter, a bent distillation adapter. We'll be using this Graham condenser. This is a type of condenser known as the Graham condenser. We have a Liebig condenser here. We'll be using this also for our extraction process. We'll be using a thermometer. Now this thermometer is connected to a rubber stopper, and we will be showing you how this rubber stopper helps us place this thermometer in the setup. And apart from that, to measure 5 grams of cinnamon leaves, we will be using a digital balance. Apart from that, we'll be using a Bunsen burner to provide heat for our extraction setup. Now that we have identified the equipment and the raw material we will be using for this experiment, we can now move on to actually carrying out this extraction process. Now students, we are going to be taking these dried cinnamon leaves and we are going to cut them into small pieces, and then we will wait, and we'll be using a round bottom flask to carry out the distillation process. So after weighing the right amount of cut pieces of cinnamon leaves, we'll be transferring them into our round bottom flask. So let's first cut these cinnamon leaves into small pieces. We will be adding the cut pieces into a petri dish. It's better if you can cut them into small pieces. Now students, we have cut a few leaves into very small pieces here, as you can see. And now we are going to measure 5 grams of these cut cinnamon leaves. So to measure the mass of these cut pieces of cinnamon leaves, we are going to be using a petri dish and a digital balance here. We will place the petri dish on the digital balance and we will use the tare function to get the reading to 0, and then we will carefully add these cut pieces of cinnamon leaves onto our petri dish here. We need to measure 5 grams of cinnamon leaves. Now we have measured exactly 5 grams of cinnamon leaves. We can transfer this amount of cinnamon leaves into our round bottom flask now and move on to adding 50 milliliters of water. Although we have measured exactly 5 grams of these cut pieces of our cinnamon leaves, we might need more than 5 grams of cinnamon leaves to obtain a substantial amount of cinnamon leaf oil. So therefore, we will be using a bit more than 5 grams in order to carry out this experiment. So now let's move on to transferring these cut pieces of cinnamon leaves into our round bottom flask. Students, now we have cut pieces of cinnamon leaves here. We have cut a few cinnamon leaves and all the cut pieces of cinnamon leaves are now present in this petri dish, and we are going to weigh the cinnamon leaves that we have here with the use of a digital balance. So we will turn on the digital balance and it reads 0 now. We are going to take an empty petri dish and we are going to place it on our digital balance, and we are going to use the tare function to get a 0 reading on the digital balance. Now, in our practical, it is advised that we take 5 grams of cinnamon leaves. However, when we are conducting this experiment, since we want to show you a considerable amount of the oil that is produced, we are going to double that amount of cinnamon leaves and get 10 grams of cinnamon leaves so that we can actually produce a larger quantity of this essential oil. So let's measure 10 grams instead of 5 grams of cinnamon leaves. Now students, we have measured 5 grams of cinnamon leaves here into this petri dish. We are going to remove it from the digital balance. Now we are going to place another petri dish here. And again, we are going to be using the tare function so that the balance reads 0. And then we are going to continue to add more cinnamon leaves to this. Now, in this petri dish also we have measured exactly 5 grams of cinnamon leaves. So together in these two petri dishes we have 10 grams of cinnamon leaves. So we can now transfer this cinnamon leaves onto our round bottom flask that we have here, and then we are going to proceed to add our water and then move on to setting up this extraction setup. So let's transfer these cut pieces of cinnamon leaves into our round bottom flask that we have here. Little by little, we can insert our cut pieces of cinnamon leaves into this round bottom flask. Whatever the cinnamon leaves that get dropped out of the flask, collect them, and you can insert them back into our round bottom flask. Like so. And after inserting all of these cinnamon leaves, we can continue to add water. So let's move on to completely filling this round bottom flask with the cut pieces of cinnamon leaves that we have measured here, and then we'll move on to the step of adding water to this round bottom flask. Now students, we have inserted all of our cut pieces of cinnamon leaves into our round bottom flask here. So let's add some water. We have our 50 milliliter measuring cylinder here, and we have some distilled water with us. So now we'll be transferring 50 milliliters of distilled water into this round bottom flask. So as you can see here, we have measured 50 milliliters of water, and we are going to be adding 50 milliliters of water into our round bottom flask here. So students, as you saw, we transferred 50 milliliters of water into this round bottom flask. But if you remember correctly, we used 10 grams of cinnamon leaves. So therefore, we should be using 100 milliliters of water. We have already transferred 50 milliliters of water into this round bottom flask. We should measure another 50 milliliters of water. We have measured another 50 milliliters of water here, and we will be transferring this 50 milliliters of water also into our round bottom flask here. Now we can move on to set up our extracting setup here, where we connect this round bottom flask into a three-way adapter, and to that three-way adapter we will be connecting our Graham condenser, and we will be setting it up so that we can extract the oil that is produced here. Now you may wonder why we use this particular setup to carry out our oil extraction. As you know, some volatiles or some essential oils that we have in different natural sources are having somewhat of a high boiling point. However, if you were to heat them so that they boil off, before boiling off, they might undergo decomposition. So when you just take the eugenol oil that you observe here and try to boil the essential oil, before they reach their boiling point, they can undergo decomposition. If you carry out this steam distillation process, however, since the essential oil is going to be mixed with water, when you have two immiscible liquids present in a mixture, the boiling point of the mixture drops below their individual boiling points. So therefore, the mixture will boil below 100 degrees Celsius, which is the boiling point of water. Therefore, we can get the essential oil to turn into a gas way below its boiling temperature, or we can say in milder conditions, we can get our essential oil to turn into a vapor. That is why we carry out this essential oil extraction process. Now students, we have included all of our cut pieces of cinnamon leaves here. Apart from that, we have added 100 milliliters of water into this round bottom flask. Now all that is left for us to do is connect this round bottom flask into our extraction setup, where we'll be connecting this through a three-way adapter and then to a Graham condenser. Now let's move on to setting up the rest of the essential oil extracting setup. Students, let's observe how we have fixed this setup to extract our essential oil. We have the round bottom flask that is filled with cut pieces of cinnamon leaves as well as 100 milliliters of water, and we can see that this round bottom flask is connected to a three-way adapter. What we have here is a three-way adapter, and on top of the three-way adapter you can see we have our thermometer fixed so that the bulb of the thermometer comes in front of this horizontal arm of the three-way adapter. So at this point, when we have the thermometer fixed in such a way, when steam is produced, we can actually measure the temperature of the steam plus the essential oil mixture by reading the thermometer reading here. So students, this setup is used for, first of all, heating the water and releasing the essential oil, and the essential oil and water vapor mixture will rise to the top, and at this point we will be reading the temperature of this gaseous vapor mixture. And with the use of the three-way adapter, the produced vapor mixture would be directed into this condenser. This condenser is also known as a Graham condenser. So this condenser will carry the gaseous mixture in this swirling glass tube, and it will carry the vapor mixture all the way to the end of the condenser that we see over here. So the gaseous mixture that is produced will be directed towards this spiraling glass tube that you see inside this Graham condenser. So this spiraling glass tube will carry the vapor mixture all the way till the end of the condenser, and it is going to be connected to this bent distill adapter, and the bent distill adapter will be directing that vapor into this separatory flask that we have over here. And this Graham condenser has a water jacket around this spiraling glass tube. So this water jacket that is going to be inside this Graham condenser is going to be cooling down the vapor as it moves from this point to the end of the condenser. So water actually will be coming in from the bottom of the condenser and will be moving up. So the water jacket will be having a water flow from this end all the way up to this end. In this way, the hottest parts of the vapor mixture comes into contact with the coldest water. So this maximizes the heat transfer. Over here, we can see when water is moving from a bottom of the condenser all the way to the top of the condenser, water is moving opposite to the direction that the vapor will be moving. This ensures that the vapor is always in contact with cold water, which maximizes the heat transfer. So this allows the vapor to cool down and turn into a liquid as the vapor mixture travels from the top of the condenser to the bottom of the condenser. So as the vapor travels, it will cool down and will turn into a liquid. So this liquid will be a mixture of two liquids, water and the essential oil extracted from the cinnamon leaves that we have over here, and this mixture will be directly poured into our separatory funnel that we have over here. So students, then later on we can use the separatory funnel to separate the two immiscible mixtures. Now, why do we use such an extraction setup to extract the essential oils that are seen in cinnamon leaves? Now, when you have essential oils such as eugenol, which are volatile essential oils, they have slightly a higher boiling point. Now, if you were to take eugenol or if you were to take any essential oil and boil them, before they reach their boiling points, they will decompose. So, if you were to extract it in such a way just by vaporizing them and then cooling them down without having this steam distillation process, before the essential oil reaches its boiling point, the essential oil will decompose. However, when you have our cinnamon leaves mixed in water here, when we are heating the mixture, due to the high temperature of the water, we can see the plant cells rupturing and releasing this essential oil into the mixture. So at higher temperatures, when the water is about to boil, you will see cinnamon leaf cells will rupture and will release the essential oils into the water that we have here. And when you continue to heat this, water and the essential oil mixture will start to boil at a lower temperature than the boiling point of both of these individual components. As you know, water boils at 100 degrees Celsius, and let's assume that the essential oil boils at a higher temperature than that. So when you have two immiscible liquids mixed together, when you're trying to boil this mixture, we have observed that the boiling temperature of this mixture will be lower than both of the individual boiling temperatures of each component. So that is why we see that this mixture will start to boil at a lower temperature than 100 degrees Celsius. This allows the essential oil to come into gaseous phase at milder conditions. So this will ensure that the essential oil that we have extracted has not undergone decomposition. So we allow the essential oil to boil at a lower temperature or a milder condition. So this essential oil that is boiled is going to be mixed with water vapor as well. So this water vapor essential oil mixture is going to rise up, and then this three-way condenser will direct this essential oil water vapor mixture over to our condenser here. And as the gaseous mixture moves from this top end of the condenser all the way to the bottom of the condenser, due to the water jacket that is present around this spiraling glass tube inside this condenser, you will see that this particular gas mixture will cool down. It will cool down below their boiling points. So therefore, this gaseous mixture will turn into a liquid. So this liquid will be collected at the end of the condenser, and this bent distilling adapter that we have connected over here will direct that liquid mixture into this separatory funnel. And later on, with the use of the separatory funnel, we will be able to separate out the oil and the water. So students, we have now observed why we use this particular setup to carry out this extraction process. And one of the questions you will be asked is why we fill in water from the bottom of this condenser and why is water removed from the top of the condenser. Now you will observe sometimes if you start filling water from the top of the condenser, due to gravity, the water will be removed from the bottom of the condenser faster, leaving some air bubbles or air gaps in the top part of the condenser, which will actually create a bit of an inefficient cooling system. Apart from that, when water is moving from the bottom all the way to the top, you will see that this condenser fills up easily, and when water is moving from the bottom of the condenser all the way to the top, the hot vapor is always in contact with cold water, making sure that the heat transfer is maximized. So students, you should know the answers to these questions when you are asked why water comes in from this end and is taken out from the top end, to maximize the heat transfer or to make sure that the hottest vapor is always in contact with the coldest water. Now we can actually move on to lighting this Bunsen burner and carrying out this essential oil extraction. Now we will be turning on the tap and we'll be allowing the water to flow from this tube, and you can see the condenser is getting filled. We started filling it from this end and water was flowing from this end all the way to the top of the condenser, and we saw that the water was filled from the bottom end of the condenser and was taken out from the top end of the condenser, and we have allowed the water to continuously run. We have not turned off the tap, so you can see water will be running from the bottom end of the condenser all the way to the top end of the condenser throughout this experiment. Now let's turn on the Bunsen burner. Now students, after lighting the Bunsen burner, you will see that the Bunsen burner will provide heat to this round bottom flask, and the water in the round bottom flask is going to heat up. As the temperature of the water increases, the cinnamon leaves that we have included in this round bottom flask will have its cells ruptured, and the essential oils in the cells that were stored in the cells will be coming out into water, like I explained earlier. So we will continue to provide heat, and at one point you will observe that there will be a vapor produced, or that this particular mixture will start to boil. So water usually boils at 100 degrees Celsius, and eugenol, which is the main essential oil found in the cinnamon leaves, has a boiling point of 254 degrees Celsius. However, when you have this oil and water mixed together, this mixture boils at a lower temperature than both of these individual components. Water boils at 100 degrees, eugenol boils at 254 degrees Celsius. However, you can see that this mixture starts to boil at a lower temperature than both of those temperatures, which means a lower temperature than even 100 degrees Celsius. So this is beneficial to us because when you are heating eugenol all the way up to 254 degrees Celsius, before they boil, it can undergo decomposition. So when we have a mixture like this boiling here, and when we have brought down the boiling point of the mixture below 100 degrees Celsius, we can ensure that the essential oil that we have over here is boiling at a lower temperature, which means the condition at which it boils is now a milder condition, meaning it will not undergo decomposition. So when this mixture is heated, the vapor mixture that is produced will be entering this condenser. As you can see, there's a water jacket around this spiraling glass tube you can see inside this condenser. So water is currently running from the bottom of the condenser all the way to the top of the condenser. So whatever the vapor that enters this condenser will cool down as it moves through the top of the condenser all the way to the bottom of the condenser. So we will continue to heat this, and you can see when the liquid is getting formed and as the gas is cooled down, you can observe a liquid getting collected at the separatory funnel we have over here. So the vapor that is produced will enter the condenser and it will cool down, will produce a liquid, and that liquid will get collected in the separatory funnel. So we'll allow this setup to get heated up for a while, and then let's come back to it when it is producing a vapor. Now students, as you can see, this mixture that we have over here is boiling. And I mentioned earlier, when this mixture is boiling, we will be producing a mixture of vapor, which is water vapor and the vapor of the essential oil. So this particular setup will start to boil at a lower temperature than both of their individual components. So the temperature at which this will start to boil is going to be lower than both the boiling temperature of water and the boiling temperature of eugenol, which is the main essential oil that is seen in cinnamon leaves. So as I mentioned, or as I observe the temperature of this particular vapor that is produced here, so we can actually measure the temperature of the vapor as we have placed this thermometer inside this three-way adapter. The temperature does not go above 99 degrees Celsius. The temperature has stopped exactly at 99 degrees Celsius. It does not go above 99 degrees Celsius or over 100 degrees Celsius. This is because, as I mentioned earlier, this produces a mix of the essential oil and water, which are two immiscible liquids. And as this mixture boils, they will boil at a lower temperature than the boiling points of each of these constituent immiscible liquids. So we can very clearly see that the boiling point is at 99 degrees Celsius here. And if you observe here, students, you can see the gas that is getting collected over here is now condensing into a liquid. Observe here, you can see the vapor that is produced, or vapor mixture that is produced, which is a mixture of the essential oil and water. So over here, the vapor that is produced is going to be a mixture of the essential oil and water. And when it is directed over here, you can see that they have condensed into a liquid. You can see that there is a translucent liquid that you can observe inside this condenser. That liquid is present there due to the condensing of the gas or vapor that was produced here. So the vapor that is produced here will be directed into this condenser, and as the condenser cools down the vapor, it will turn into a liquid, as you can see here. And the liquid will slowly move into the bottom of the condenser here, and it will be poured out of the condenser into this separatory funnel. And we have made sure that the separatory funnel is tightly closed, so the liquid that is produced will get directly collected into our separatory funnel. Now students, as you can see, when we keep heating this mixture, the temperature still remains at 99 degrees Celsius. It doesn't go above 99 degrees Celsius. And we can see the liquid is slowly getting collected into this separatory funnel we have over here. So as the vapor enters into the condenser, condenser will cool down the vapor, and little by little you can see the vapor will turn into a liquid and will get collected over to our separatory funnel. You can very clearly observe the condensed vapor in this area, and it is filling up the rest of the condenser as well. So slowly you can see now again a small amount of liquid got collected over here onto the separatory funnel. Please observe this condenser. You can see how it is slowly moving the liquid over to the bottom of the condenser and out of the condenser into the separatory funnel. Students, we will allow this setup to run for a few more minutes, and after we have collected a sufficient amount of this liquid mixture, we are going to move on into the next part of this experiment. Now students, we can see that the water that is present inside this round bottom flask is almost over, meaning majority of the water that was taken into our round bottom flask has turned into a gas, and the temperature over here is still at 99 degrees Celsius, indicating that there is still the essential oil and water mixture present here. If all of the essential oil is completely gone, if there's only water, the temperature will be at 100 degrees Celsius. However, after we observe that most of the water has gotten evaporated, we are going to stop heating this mixture, and whatever the remaining amount of the liquid that is present here in the condenser will be collected into our separatory funnel. The liquid that you see over here is a mixture of water and the essential oil. As we heat this up, both water vapor and essential oil vapor will be coming into the condenser, and when they reach a temperature way below their boiling points, they will turn into a liquid, as you can see here. So this liquid should be collected into our separatory funnel after we take the heat source away from the round bottom flask. So you will gradually turn it off and take off the heat source from the round bottom flask. If you quickly do it, the pressure inside the system will drop, and some of this water and essential oil mixture will run back into the round bottom flask. To avoid that from happening, we will gradually decrease the temperature, gradually cool down the system, and we will remove the heat source from this round bottom flask. So we will come again to meet you after we have separated the heat source from this setup, and after we have collected all of this liquid mixture into our separatory flask. Now students, we have extracted an essential oil from cinnamon leaves. You saw how the extraction setup was fixed, you saw how it was carried out, and now we have the extraction over here. So this contains water mixed with the eugenol oil or essential oil that we extracted with the help of cinnamon leaves. So over here, the essential oil and water is mixed. Essential oil, in this case, since we used cinnamon leaves, the main component of the essential oil that we have here is eugenol. So this particular essential oil is not very soluble in water. That's why you can observe this cloudiness in this aqueous mixture. However, if you introduce a separate organic solvent that can dissolve this oil in the solvent better, what happens is, if you mix organic solvents such as diethyl ether that we have here with this aqueous mixture, you will observe that majority of the oil that we have here dispersed throughout this aqueous medium will easily dissolve in the diethyl ether organic solvent. So diethyl ether is going to be a nonpolar solvent, and this nonpolar eugenol or the essential oil that we have produced here will dissolve in our diethyl ether. So through this solvent extraction process, we can actually extract the oil that is now mixed in water to an organic solvent such as diethyl ether. So now we are going to be moving on to that step where we will be measuring 10 cubic centimeters of diethyl ether, and we will transfer it into this separating funnel, and then we will mix the solution really well. And after mixing the solution, we are going to be separating the diethyl ether layer from this aqueous mixture. So majority of the oil that is present in this aqueous mixture will dissolve in the diethyl ether layer, and we will continue to do this multiple times until almost all of the oil or essential oil that is present in this aqueous mixture is extracted onto our organic solvent layer. So let's move ahead and do that. So we have our measuring cylinder here. We are going to measure 10 milliliters of diethyl ether and transfer that into our separating funnel. We'll be measuring 10 cubic centimeters or 10 milliliters of diethyl ether. Now we'll be transferring this 10 milliliters of diethyl ether into our separating funnel. After transferring it, we can close the separating funnel like so, and we will be shaking it so that it gets mixed really well. And students, don't forget to relieve the pressure every now and again. So after mixing it really well, you can open this stopcock and release the pressure every time that you mix it. You can continue to shake it, and then you can relieve the pressure by opening the lid. If you prefer to keep it upside down, you can shake it and then after that open the stopcock to release the pressure. But if you prefer opening the lid instead, you can proceed to do so. After mixing it a few times, you can then place it on our retort stand here and allow the two layers to separate. And after the two layers have separated, we can remove our organic solvent layer with the use of this separating funnel. And then we will repeat this process 2 more times to extract the most amount of essential oil from this aqueous mixture. Now students, we have allowed this mixture to settle for a while so that the two layers will separate. Now it is time for us to separate the layers. So we will take the first layer into a beaker, because the first layer is going to be the aqueous layer with where the essential oil is mixed. So we can see over here the bottom layer is going to be your aqueous layer where the essential oil is mixed in the aqueous layer. You can see earlier on, before we added the diethyl ether, the aqueous layer was very cloudy in nature. But after we added the diethyl ether and mixed it with the diethyl ether, we saw that the aqueous layer got slightly clearer. This is because the immiscible oil that was mixed in the aqueous layer is now dissolved in the organic solvent that we added, which is diethyl ether. So as it is dissolved in the diethyl ether, you can see the aqueous layer is starting to clear up. And since we added very small amount of diethyl ether, we have a very small layer of diethyl ether at the top of this aqueous mixture. You can't really see it now, but when we reduce the amount of aqueous layer by opening this stopcock, you will see as the aqueous layer gets collected to this beaker, around this area you will be able to very clearly see the diethyl ether layer. And now let's turn this stopcock and allow the aqueous layer to get collected into the beaker. But students, as you turn this on, please make sure to lift this lid, otherwise the water or the aqueous layer will not be running down through this. Otherwise the aqueous layer will not run down. Now you can see the aqueous layer is getting collected into this beaker, and as you can see, when the water level or when the liquid level is going down, we can very clearly see the diethyl ether layer over here. That's a very small diethyl ether layer that can be observed over here. We can now very clearly see the two separate layers. There's a very small layer that we can observe over here. That is going to be our diethyl ether layer. So students, as you can see, the diethyl ether layer is up on top here and the aqueous layer is in the lower parts over here. Diethyl ether layer or diethyl ether has a lower density than water, so it is going to be present on the top parts of this mixture. It doesn't mix well with water, so that's why they separate out into two separate layers. And since diethyl ether layer has a lower density, it is going to be found on the top layer in this mixture. Now we will very carefully separate out the two layers by turning this stopcock and allowing only the water layer to run down. And as soon as we have removed all of the water, now students, we have stopped collecting the aqueous layer into this beaker, because what is remaining in the separating funnel is now just our diethyl ether layer. So we are going to now place our Erlenmeyer flask and collect our diethyl ether layer into our Erlenmeyer flask, like so. And after collecting our diethyl ether layer, we are going to place a rubber stopper to make sure that it doesn't get evaporated. So we have collected our diethyl ether layer here. We will place it aside, and we will continue to repeat this process 2 more times with the use of diethyl ether. Now we will add the aqueous mixture again into our separating funnel. Before we do that, we can make sure that the stopcock is perfectly closed. After that, we can proceed to add this aqueous mixture into our separating funnel. Now, after taking our aqueous mixture into the separating funnel again, we are going to add 10 cubic centimeters of our diethyl ether into this one more time. We are going to measure 10 cubic centimeters of diethyl ether. Now, we'll transfer this 10 cubic centimeters of diethyl ether into our aqueous mixture that we have here. After adding the 10 cubic centimeters of diethyl ether, we'll continue to shake it, but when you're shaking it, please make sure to close the stopcock tightly and then mix it really well. After that, to relieve the pressure, you can open the stopcock and release the pressure that is built inside the separating funnel. You should not allow the pressure to build up inside the separating funnel for too long. Now we have mixed it really well. We will now place this in the stand and we'll allow it to settle down and separate out into two layers. After it separates into two layers, we will be using the stopcock again to separate the aqueous layer from our organic solvent layer, in this case from our diethyl ether layer. So let's wait for a few minutes and then let's remove the aqueous layer into this beaker and the diethyl ether layer into our Erlenmeyer flask we have here. Now students, we have allowed this mixture to settle for a while, so we can observe two layers. Over here we have a very small diethyl ether layer, and at the bottom you can see an aqueous layer, and you can very clearly see that the aqueous layer is now very clear. This is because almost all of the essential oil is now dissolved in the diethyl ether layer. At the bottom what we have is our aqueous layer, and at the top we have our diethyl ether layer. So let's open the stopcock and allow the aqueous layer to get collected into our beaker that we have over here. Now at this point you can observe there is two layers here. The top layer that you can see over here is your diethyl ether layer, and the bottom layer is your aqueous layer. So I hope you can very clearly see those two layers here, and the aqueous layer is now very clear due to majority of the essential oil dissolving in our diethyl ether layer. So let's proceed to separate them out. Now we have very carefully separated out our aqueous layer. We can now take our Erlenmeyer flask that we collected our previous diethyl ether layer to, and we can allow this diethyl ether layer to get collected into the same Erlenmeyer flask where we collected our previous diethyl ether layer too. Now you can see we have two 10 milliliter portions of our diethyl ether layer collected here in this Erlenmeyer flask. We will place a rubber stopper on this Erlenmeyer flask so that the diethyl ether that we have taken into this flask won't evaporate as we carry out another solvent extraction. So to carry out our third and last solvent extraction, we will again tighten our stopcock in this separating funnel that we have here, and we will transfer this aqueous mixture into our separating funnel, and afterwards we will measure 10 milliliters of diethyl ether like we did in the previous two instances. Now we have measured 10 cubic centimeters of diethyl ether, and we can now proceed to mix this, and we can turn the stopcock to relieve the pressure like I have taught earlier. We can mix it really well, and then we can turn the stopcock to release the pressure like we have done in the previous two instances. We will repeat this a couple of times. After mixing the mixture quite well a couple of times like this, we can then place this separating funnel in the stand, and we can allow the mixture to separate into two layers within a matter of few minutes. And after it separates into two layers, we can proceed to separate the two layers out with the use of our stopcock that we have over here. So we will allow this mixture to separate out into two layers, and then we can proceed to separate out the aqueous layer into this beaker and the diethyl ether layer into our Erlenmeyer flask we have here. Now we will proceed to separate the aqueous layer. As you can see, we can now very clearly see the two layers, the diethyl ether layer on top and the aqueous layer on the bottom. We will separate out the two layers very carefully. Now we have very carefully separated out only the aqueous layer into this beaker, and we can take our Erlenmeyer flask and continue to separate out our diethyl ether layer into our Erlenmeyer flask. Now students, what we have over here is our diethyl ether mixed with our essential oil. There might be traces of water here as well. When we are separating the diethyl ether layer from the water layer, we might have taken a small amount of water along with the diethyl ether layer. So it is a requirement for us to remove these excess traces of water from this diethyl ether layer that we have over here. In order to do that, we can use a drying agent. Over here we have a drying agent, sodium sulfate, or more accurately we have anhydrous sodium sulfate in front of us here. So we are going to be taking this anhydrous sodium sulfate and we will transfer a small amount of the solid into this diethyl ether layer. The reason why we have closed this with a stopper, as I mentioned earlier, is because diethyl ether is very volatile and it might get evaporated if we keep it without placing a stopper on the Erlenmeyer flask. So when we add sodium sulfate to our diethyl ether layer that we have separated out, if it contains traces of water, we can see this sodium sulfate clumping together. If you keep on swirling it for a while and if you add a little bit more sodium sulfate, there will come a time where the sodium sulfate no longer clumps together. At that point, you can be sure that the diethyl ether layer is no longer containing any traces of water. So now let's proceed to add a small amount of sodium sulfate into this and observe whether we have some water present in our diethyl ether layer. We will take a small amount of sodium sulfate and we can add it to our, now we can add this sodium sulfate into our diethyl ether layer. We will mix it for a while to see if the sodium sulfate is clumping together. Now students, as you can see, when we are swirling this around, we can't see the sodium sulfate clumping together. This is indicating that we do not have excess water present in the medium, but just to be sure, we can add a few more scoops of sodium sulfate. And we can see the sodium sulfate is not clumping together. This is indicating that there are no traces of water present in this diethyl ether layer. You do not see any clumps of sodium sulfate formed here. You can see all of the sodium sulfate is dispersed and they are not clumping together. As you can see here students, the sodium sulfate is not clumping together when swirled. This indicates that there is no traces of water additionally found in this diethyl ether layer. Now we can move on to the next step of this process. Now students, we have a distillation setup that is fixed in front of us. We can see we have fixed a distilling flask, connected it to a Liebig condenser here, and at the end of a Liebig condenser we have a conical flask that is set up to collect a fraction of the liquid that is separated from this mixture of liquids we will have in this round bottom flask. So to this distilling flask we are going to be adding our diethyl ether layer that we extracted from the solvent extraction process. So this diethyl ether has now the essential oil dissolved in it. Essential oil was more soluble in the diethyl ether layer than in aqueous layer. So we saw when we were carrying out the solvent extraction process, we saw that the essential oil that was released into the aqueous layer was dissolved more in the diethyl ether layer, and with the use of a separating funnel, we were able to separate out the aqueous layer and the diethyl ether layer separately. And we actually carried out solvent extraction 3 times to make sure that all of the essential oil that is present in the aqueous medium was removed onto our diethyl ether layer. So the essential oil that was originally present in the leaves of cinnamon is now extracted into this diethyl ether layer we have in front of us. So still, this is 2 main things. We have diethyl ether in this Erlenmeyer flask mixed with our essential oil. They are very miscible with one another because they are dissolving in one another very well. So how are we going to separate these 2 out? We can use simple distillation. So when we are carrying out this simple distillation, the component with a lower boiling point will boil first. In this case, diethyl ether will boil first and will turn into a gas, and it will be removed from the mixture as a gas. So it will turn into a gas and it will be directed onto this Liebig condenser, and in the Liebig condenser it will cool down to produce a liquid, and it will be collected in this Erlenmeyer flask. And in this distilling flask we have over here, you will see the component which has a higher boiling point remaining here. The component that had the higher boiling point was the essential oil, which had a boiling point of around 254 degrees Celsius. Eugenol had a higher boiling point. Since we are using a steam bath to heat this, the temperature of the system will never increase enough to boil out eugenol. Since the heat is provided through steam, the highest temperature that you can observe within this distilling flask would be 100 degrees Celsius. And that temperature is enough to boil our diethyl ether layer. However, it is not a temperature that is high enough to boil our essential oil that we have here. So you will see at the end of this experiment, the essential oil will be contained in this distilling flask. So let's add our essential oil and diethyl ether mixture that we have over here. You can see the sodium sulfate that we added earlier. It is not clumping together. When we mix it, we can see it is not clumping together still. It has been quite some time now. It is not clumping together. This is indicative of the fact that all of the water in the mixture is now removed. So we can proceed to decant this very carefully and add our diethyl ether layer slowly into this distilling flask. And we'll be adding it into this distilling flask, making sure the sodium sulfate does not get transferred into our distilling flask. Now students, you can see the sodium sulfate did not get transferred into our distilling flask. However, all of the diethyl ether got transferred into our distilling flask. So diethyl ether has dissolved essential oils in it. You can see as this system heats up, you will see the steam that is produced will heat up this diethyl ether layer that we have here, and all of the diethyl ether will get evaporated and will be separated out into this titration flask we have here. Now students, as the system is starting to produce steam, we will turn on our tap to fill this Liebig condenser. So let's go ahead and turn the tap on. You can see that this particular Liebig condenser is completely filled now. What is entering the Liebig condenser from this end and is traveling all the way up to this end. Now students, as we heat this beaker containing water, steam will be produced, and as you can see, steam is produced here, and that steam is heating up this distillation flask and the contents in it. And as I mentioned earlier, the diethyl ether layer is going to get evaporated, leaving behind the essential oil, and we can see the diethyl ether layer has gotten smaller in volume here. That means some vapor has been produced, and that vapor will be directed towards the Liebig condenser, and we can see some amount of diethyl ether layer has gotten condensed and has come into this titration flask. So as time goes by, we will see only the essential oil remaining over here. So we will allow the system to run for a while, and we will come back and observe the essential oil remaining in our distillation flask. Now students, we have allowed this system to run for a few minutes. Now we have evaporated our diethyl ether layer, and we only have the essential oil remaining over here. So as we use cinnamon leaves, the main component found in the essential oil of cinnamon leaves is going to be eugenol. So what we see here is eugenol that is remaining on our distilling flask. So to see it very clearly, let's remove this steam bath from this setup, and we'll come back to observe the essential oil that is remaining in our distillation flask. Now students, after removing the steam bath, you can very clearly see the remaining amount of the essential oil that we extracted. So you can see, using 10 grams of cinnamon leaves, we could only extract a very small amount of this essential oil. We can observe this essential oil, since it's at the bottom of the distillation flask here. Since we can't very clearly see it, we'll try to get it to a dropper here to clearly observe its color. So we can see here, this is the amount of essential oil that we extracted. And right now, the entire area is surrounded with the pleasant smell or pleasant odor coming from this essential oil. So we have now extracted a small portion of cinnamon leaf oil, or in other words, eugenol essential oil over here. As we were carrying out the simple distillation process, to this distilling flask, we added a diethyl ether layer that had this essential oil dissolved in it. So as we were heating this distilling flask, we saw the diethyl ether was getting evaporated and was directed towards this Liebig condenser, and it condensed the diethyl ether, and a small portion of this diethyl ether that was produced is collected over here and is present in this Erlenmeyer flask. Majority of the diethyl ether was evaporated as it is very volatile. So even after getting it collected to this Erlenmeyer flask, you would observe that the diethyl ether quickly evaporates and only a very small amount is still remaining in our Erlenmeyer flask. So in this particular experiment, we looked at how to extract an essential oil from a plant source. Specifically, we used cinnamon leaves. Depending on which part of the cinnamon plant we extract the oil, we will get different types of oils. since we used cinnamon leaves, we extracted eugenol essential oil. When we were starting this experiment, we cut the cinnamon leaves into small pieces and we added it to a round bottom flask, and then we added water to it and we directly heated the round bottom flask. As we directly heated the round bottom flask, the water got heated up, and along with it, the pieces of cinnamon leaves also got heated. This allowed the cells of the cinnamon leaves to get ruptured, and the essential oils which were present inside the cell was released onto the water that was in the round bottom flask. The essential oil and water are not miscible. They are not soluble in one another. So when you have two immiscible liquids mixed together, we learned that their boiling point, or the boiling point of the mixture, will come down below each of their individual boiling points. Water would usually boil at 100 degrees Celsius, and when it comes to eugenol that we observe here, it would normally boil at 254 degrees Celsius. But this mixture, we saw when we were carrying out this experiment, we saw this mixture was getting boiled at 99 degrees Celsius, below both of their boiling points. So this particular method of extracting essential oils is beneficial to us because you will not need higher temperatures to extract the essential oil. Because when you're trying to boil essential oil on its own by increasing the temperature all the way up to its boiling point, majority of the time the essential oil will get decomposed before reaching its boiling point. However, when the essential oil is mixed with water, since these two are immiscible with one another, the boiling point of the mixture is brought down to a lower temperature. So therefore, we can boil the essential oil at a lower temperature, since the entire mixture will boil at a temperature lower than both of the constituent liquids' boiling points. So after we boil the mixture, the vapor that was produced was condensed using a Graham condenser. So the condensed vapor contained both water and the essential oil, and we saw how it got collected into a separation flask. So in the separating flask, we saw the mixture that was obtained was cloudy in nature and had a white, cloudy color to it. However, when a separate solvent, which is diethyl ether, was added to this aqueous mixture, we saw the cloudy nature of the aqueous mixture disappeared. The cloudy nature of the aqueous mixture was present due to the essential oil being mixed in the aqueous medium, and since they were not very miscible with one another, you could observe that cloudy nature. When we added diethyl ether, majority of the essential oil got dissolved in the diethyl ether layer, and the water layer started clearing out. And this is referred to as solvent extraction method. So through the solvent extraction method, majority of the essential oil that was present in water was extracted to diethyl ether layer. This extraction was only possible because the essential oil was more soluble in diethyl ether than in water. That is why the essential oil got dissolved more in the diethyl ether layer. Afterwards, we extracted the diethyl ether layer through the separating funnel, and we added sodium sulfate, anhydrous sodium sulfate, to remove any traces of water that was found in the diethyl ether layer. And if there were any water in the diethyl ether layer, when you add anhydrous sodium sulfate, when you keep swirling the mixture around, you would see the formation of sodium sulfate clumps. Rather than sodium sulfate being mixed throughout the mixture, you will see the formation of sodium sulfate clumps. If you do not observe the formation of any sodium sulfate clumps, this indicates that your diethyl ether layer is now free of trace amounts of water. So after removing all water, we use this diethyl ether layer that contains the essential oil and we transferred it into this distilling flask. And we carried out a simple distillation over here. And as we carried out the simple distillation, we saw the component out of the two components that has the lower boiling point got boiled off first. So this particular mixture had diethyl ether, which has a boiling point of somewhere around 34 to 35 degrees Celsius, and it also contained the essential oil eugenol, which has a boiling point of 254 degrees Celsius. So the first thing to boil off when provided with heat to this mixture would be diethyl ether. And as we predicted, the diethyl ether got evaporated, leaving behind the essential oil. And the evaporated diethyl ether was condensed with the use of a Liebig condenser and was collected to this Erlenmeyer flask. And in this distilling flask, we have our essential oil remaining, which happens to be eugenol. We have now observed how this essential oil was extracted from cinnamon leaves. Now we can move on to a small additional part to explain how the dehydration process was done with the use of anhydrous sodium sulfate. Now students, let's observe how sodium sulfate will appear if traces of water was actually present in a given sample where water was not supposed to be found. So to observe how sodium sulfate absorbs water and clumps together, we can use a beaker and add a small amount of water into this beaker with the use of a wash bottle. And to this beaker, we are going to add a small amount of sodium sulfate. And you will see in the presence of water how sodium sulfate will clump together as we add sodium sulfate to water and we start to mix it. You can see sodium sulfate clumps together, and this is a clear indication that sodium sulfate has come into contact with water. Then we will move on to doing the same with methanol, and we will observe whether the methanol sample we have here has any water in it. So let's first take sodium sulfate bottle we have here, and we will add a small amount of sodium sulfate, a white color solid, into water. We'll add 2 spatulas full of sodium sulfate into this water sample, and let's mix it really well. As you mix it, you will start to observe that the sodium sulfate is forming clumps of sodium sulfate. Students, can you see that the sodium sulfate is clumping together? Now, right now, after I mixed it, it is not very clear to observe. We'll allow this to settle for a while and we'll come back to it and we'll observe how the sodium sulfate actually is not found as a powder anymore. It is actually clumped together in this aqueous medium after coming into contact with water. Now students, you can see how the sodium sulfate has gotten clumped together when it has come into contact with water. So some of the sodium sulfate has dissolved in water, but the rest of it is now forming clumps of sodium sulfate solid. So when we added sodium sulfate into water, after they come into contact with water, we can see how the sodium sulfate is forming clumps of sodium sulfate rather than being present as a powder in aqueous medium. Now, let's do the same to a small volume of methanol. We have our methanol here. Let's take a small amount of methanol into our beaker. And let's add a spatula full of sodium sulfate and observe whether it forms clumps in methanol. We will mix it with a stirring rod as well. As you can see, students, when mixed in methanol, the sodium sulfate does not form clumps of sodium sulfate. Over here, we can very clearly see the formation of clumps. You can see sodium sulfate is present as clumps of sodium sulfate. But if you observe over here, the sodium sulfate is present as a powder. If you mix it really well, you can see sodium sulfate is not forming clumps. There are no clumps observed when you mix sodium sulfate in the methanol that we have here. You do not observe any clumps formed when we have added sodium sulfate to methanol. It falls like powder inside the methanol without forming any clumps. However, in water, you can very clearly see the clumps that are observed here. It is not present as a powder. When you mix it, you can very clearly see it is not present as a powder. It is present as one big clump of sodium sulfate. When it comes to methanol, it is not present as a clump. You can see the sodium sulfate is present as a powder. So this very clearly gives you an idea how we can figure out when sodium sulfate is added to an organic layer. If sodium sulfate forms clumps of sodium sulfate, that indicates that the organic solvent layer has had some water in it. If you continuously add sodium sulfate, you will come to a point where sodium sulfate no longer forms any sodium sulfate clumps. The excess or the added sodium sulfate is going to remain as a powder without forming clumps. At this point, you can be sure that all the water in the sample has been taken away by reacting with sodium sulfate. So using sodium hydrate, we can remove the water that is found in an organic solvent layer. That is why sodium sulfate is referred to as a drying agent in this part of our experiment. So students, I hope you have developed an idea why we use sodium sulfate right after our solvent extraction process. Right after the solvent extraction process, to the diethyl ether layer, we added sodium sulfate to remove any trace amounts of water that might have gotten mixed with the diethyl ether layer. And by adding sodium sulfate, after removing any trace amounts of water that was found in the diethyl ether layer, we moved on to the simple distillation process, making sure no water was found in the diethyl ether layer. So this additional part of our experiment was conducted to give you a very clear idea of what happens when sodium sulfate is included in our organic layer, and if water is present in the organic layer, what the observation would be, and if no water is found in the organic layer, what the observation would be, was clearly discussed in this part of our experiment. Now students, we are back in our classroom and we are going to be taking a look at some questions that are associated with our experiment number 42. So let's take a look at the first question. So first question asks us to briefly explain why steam distillation, explain why steam distillation can be conducted at a lower temperature than the boiling points of both pure essential oils and water when extracting essential oils from plant materials. So when we are carrying out an extraction of a particular essential oil from plant material, students, we can carry out steam distillation. And we just looked at the practical. But at the practical also we noticed that this extraction or steam distillation will take place at a lower temperature compared to both the boiling temperature of essential oil as well as the boiling temperature of water. Now, why is this happening? This is happening because when you are trying to take a look, let's take a look at what is taking place here. So in this case, we have water, and then we will have some plant material. Let's say we have some leaves here, right? Now students, when we are heating this, and even without heating, in this area, we will have water vapor. We will have water vapor. And apart from that, if we use a different color for the vapor of the essential oil, because of the heat, the essential oil will also easily vaporize. So let's say we have the vapor of the essential oil also here. We will indicate it in red like this, right? So both of these vapors will be present here when we are heating the mixture. Essential oil vapor coming from these leaves, right? And water vapor coming from the solvent water here. So now students, usually when a liquid boils, now we'll quickly turn to boiling a liquid. So if you are talking about boiling a liquid, if you just take only water, and when you're trying to boil this liquid, if you just have water here, liquid water, right? So over here we will have, if you take water, always water or any solvent, we know is always in equilibrium with its liquid state and the gaseous state. So here we have water vapor, we'll say H2O gas, and we do know that there is an equilibrium between them, right? H2O liquid will turn into H2O gas and gas into liquid. So this is an equilibrium that is taking place, right? So this is H2O gas or vapor. And at different temperatures, students, the amount of vapor that exists in this area will vary. At a lower temperature, you will have a small amount of vapor here, and at a particular temperature, when that region is completely saturated with a given vapor, we call that area to be having a saturated vapor pressure. Because of the vapor, because of the gas that is present there, there will be a pressure. So when this area is saturated with water vapor, here we are going to have something referred to as a saturated vapor pressure, right? So we'll call that pressure P naught H2O, right? So now students, this particular saturated vapor pressure will increase as we increase the temperature, because at higher temperatures the vapor that is present over here will increase, and therefore the pressure that they generate will also increase, and because of that you will have a higher saturated vapor pressure when you increase the temperature. So although we have a bit of a pressure generated through this vapor, we shouldn't forget that there's something called atmospheric pressure. So this atmospheric pressure is pushing down this vapor and is applied on this entire system. So this is your atmospheric pressure. Let's say P atmosphere. So at one point, students, this saturated vapor pressure, when you keep on heating this water, when you keep on heating this water, at one point this saturated vapor pressure is going to be equal to the external atmospheric pressure. So atmospheric pressure is, let's imagine this, the atmospheric is pushing everything down, but the saturated vapor pressure is, let's say, exerting a pressure in the other direction. So basically, students, when the time at which the saturated vapor pressure becomes equivalent to the atmospheric pressure, these two pressures will be having the same magnitude, so they cancel out. So at that point there will be no pressure that keeping this water in the liquid state. We say they turn into gases, and you can actually observe this because there will be water bubbles or air bubbles formed within the water volume here, and you can see the water starting to boil, right? So whenever we want to boil a liquid, what is essentially happening is we are increasing the temperature of the liquid until its vapor pressure, saturated vapor pressure, is going to be equal to the atmospheric pressure, right? That's why when we use a pressure cooker, water boils at a higher temperature, because within the pressure cooker, in the upper area, the pressure is high. So water is not necessarily boiling at a lower temperature. Water will require a higher temperature to boil. So that's why food gets cooked a bit better, a bit faster, it becomes softer, because now the water is at a higher temperature, because otherwise water can only reach 100 degrees Celsius temperature at usual atmospheric pressure, but inside a pressure cooker they can reach high temperatures, right? Now over here, students, when you are leaving a system open to the atmospheric pressure, as soon as the saturated vapor pressure reaches the value of atmospheric pressure, then the liquid will start to boil. So the requirement here for a liquid to boil is saturated vapor pressure being equal to atmospheric pressure. But in the steam distillation process, we have not just water, we have plant material containing essential oils also. So when you are providing heat, where you can observe in the vapor region here, you have both water vapor as well as our essential oils vapor. So there are two types of vapors that we find here: vapor of the essential oil as well as the vapor of the water. So in this case, we don't have to wait till, now this contains, let's say, PH2O gas, right? Our vapor pressure of water is present here, as well as P, let's say, essential oil. I'll write EO, right? So in this vapor region, both of these vapors are present and they are saturated here. And at this point, students, the total vapor pressure comes from both of these vapors, the total vapor pressure. So when the total vapor pressure is
equal to atmospheric pressure, the liquid will start to boil. So in this particular scenario, you don't have to wait until one of these vapor pressures reaches atmospheric pressure. You don't have to wait till one of these vapor pressures reach atmospheric pressure. We have to only wait the cumulative or the total vapor pressure reaches atmospheric pressure. So neither one of these vapor pressures ever have to individually reach 1 atm atmospheric pressure. They have to only together reach to 1 atm pressure, right? So because of this, students, the boiling temperature of this mixture would be happening at a lower temperature than their individual boiling temperatures. Is that clear? So when it comes to these boiling temperature related question, you have to state that when a liquid boils, you have to mention when a liquid boils, the saturated vapor pressure should be equal to the atmospheric pressure. So atmospheric pressure is 1 atm. So you can say that the saturated vapor pressure should be equal to the atmospheric pressure 1 atm. So at that point, a liquid will boil. But when it comes to our distillation process, we have water as well as a plant material containing an essential oil. So when we are heating this mixture, in the vapor region, you not only have vapor pressure of water, but also the essential oils vapor pressure will also be there. So you have to write in the vapor region or region above the aqueous medium will contain both H2O vapor as well as essential oil vapor, and they are going to saturate this region. So in this scenario, the saturated vapor pressure of water plus the saturated vapor pressure of essential oil is considered to be the total vapor pressure. And when the total vapor pressure is going to be equal to atmospheric pressure, the mixture will start to boil. Since each of these vapor pressures do not have to reach 1 atm pressure individually, we don't have to heat the system up all the way to their individual boiling points. Instead, at the point at which the total vapor pressure, that is the addition of essential oils saturated vapor pressure as well as water saturated vapor pressure. So when the total vapor pressure becomes equal to atmospheric pressure, we can say the boiling takes place. So therefore, steam distillation can be done at that temperature. So you can also indicate the total pressure PT, right? Total vapor pressure is equal to vapor pressure coming from water, saturated vapor pressure, plus P naught essential oil vapor pressure. We can write when PT equals P atmosphere, the liquid will start to boil. So since we are only waiting till the PT to reach this atmospheric pressure, we will have the liquid boiling before each of these boiling points are reached individually. So when it comes to water's boiling point, it's 100 degrees Celsius. So we don't have to wait till 100 degrees Celsius to see this solution or see this mixture boil, right? Because what we are looking at is the overall pressure that is generated by saturated vapor pressure of water as well as saturated vapor pressure of essential oil. So if you have written an answer including all of these points, you will get your marks. So that is the answer to this question. You have to make sure to state each of these points. If you take each of these water and essential oil separately into 2 beakers for you to boil them, they have to individually have their boiling points reached when their saturated vapor pressures are equal to 180 atm or atmospheric pressure. But here they don't have to individually reach 180 atm, they have to collectively reach 180. So their total pressure should reach 180 atm pressure. So at that point the mixture will start to boil. If they were alone as a pure substance, like let's say pure water and pure essential oil, they have to be heated until their own individual vapor pressure reaches 180 atm. So because of that, sometimes you can see water, of course, has a bit of a higher boiling point, because for you to reach 180 atm when it comes to water saturated vapor pressure, you have to heat the water up to 100 degrees Celsius. But here we do not have to do that, because neither water vapor pressure or the essential oil vapor pressure neither of them have to reach 180 atm. They have to collectively reach 180 atm. When they are added together, only the 180 atm value should be obtained. Right, so that is why the boiling takes place before reaching either one of their boiling points. So in this part they are asking us, what are 3 types of plant material commonly utilized in Sri Lanka for extracting essential oils, and what are primary components found in these essential oils? So maybe we can have a small table here so that we can easily list down the different types of essential oils we are dealing with. So they are asking only for 3. So let's write 3 different types of essential oils that we come across. Right. Right. So now we will write the name of the plant here, plant, and they're asking for the primary component or main components. Main component in the essential oil. So the first thing that we think of whenever they're asking about essential oils is the cinnamon plant. You know, in Sri Lanka we do grow cinnamon. Actually, we provide cinnamon to the international market, and we are one of the largest suppliers there is when it comes to cinnamon. So we take cinnamon from cinnamon oil from cinnamon plant. So we do have, even within the country, a lot of people are circulating these different types of cinnamon oils. So when it comes to cinnamon oil, students, always check. There are 2 main types of cinnamon oils. Depending on your preference, you might want one or the other. So one type of oil is extracted from cinnamon leaves. These are relatively inexpensive. The other type of oil is extracted from cinnamon bark, which is going to be relatively bit more expensive. So when it comes to cinnamon, we use both leaves, right? We use the leaves, and we use the bark as well. We use the bark as well. Use both the leaves and the barks. So leaves usually has the cinnamon oil that is extracted from leaves. The main component is eugenol. And when it comes to bark, that is cinnamaldehyde. Cinnamaldehyde. And apart from that, students, we use other different types of plant material as well. One is clove. We use clove to extract clove oil. So there also the main component is in clove oil, the main component is eugenol. The main component is eugenol. Apart from that, we use different types of plants known as pangiri. So we have Heen pangiri, Maha pangiri, and Lenabatu pangiri. These names are actually given in our textbook as well. You can actually go through and come across these names. So these oils or these plants can be used to extract citronella oil. Citronella oil. So in this pangiri plants, the oil that is extracted is citronella oil, and in citronella oil you have the main component citronellal. Apart from that, we can also talk about lemongrass. Lemongrass, also in Sinhalese known as sera. So lemongrass can also be used to extract an essential oil. It mainly contains citral. Mainly contains citral. Right. So these are the main different types of plant material that you can use, as well as the main components of the essential oils. So these are the main types of plants that we are using to extract different types of essential oils, and these are the main components found in each of these essential oils that we extract. So that is the answer to the question 1b. Let's move on to 1c. List 3 uses of each of these essential oils mentioned in part B above. So now we have to list 3 uses of each of these essential oils that we have mentioned above. So let's go on and mention the uses here. We can start with eugenol. So eugenol is used when it comes to manufacturing toothpaste, in herbal medication, in herbal medicines. Apart from that, this particular eugenol essential oil can also be used as a flavoring agent, right? So it is used as a flavoring agent. Right, so these are some of its uses. And for the fourth use, we can also say that these are used in cosmetic products or cosmetic products. So apart from that, now we have spoken about eugenol, we have cinnamaldehyde, again eugenol here, and then we have pangiri citronelle. Actually, when it comes to citronella, more than citronellal, we actually have something called geraniol. So we have to include that as well. Right, so geraniol is another component that is that is more prominently there than citronellal. Actually, the geraniol percentage is a bit higher. Geraniol percentage is slightly higher. right? So when it comes to citronella oil, geraniol is actually going to be the major component. More than citronella, geraniol percentage is higher in citronella oil. So now we discussed eugenol, and then we can move on to, we can see eugenol is present in both cinnamon leaf oil as well as clove oil, and then we can move on to we'll move on to geraniol. We'll move on to geraniol. It is used in cosmetics. It is used as an insect repellent. You would have heard about citronella oils being used to repel mosquitoes, right? So apart from that, we can also use this citronella oil or geraniol for as a fungicide. So these are three uses, right? In medicines also we can use this. So if you include, when you're producing medicine and as a flavoring agent, we can use this. Apart from that, we also have to speak about another component here, which is citral. We'll talk about citral here. Again, we use citral when it comes to flavoring. As a in cosmetic products, you find this. When it comes to producing perfumes, also we use this. So these are three uses. If you have written three, that will be sufficient. So apart from these three, you can see these things in medicines as well, especially in herbal medication or Ayurvedic medicine. And apart from that, we have cinnamaldehyde. We have cinnamaldehyde. So we use this in food actually as a flavoring, right? As a flavoring. You can find these in cosmetics. And in toothpaste as well. So these are some uses, not limited to only these, but definitely these are some uses that are revolving around these essential oils, or the main components of the essential oils that are listed here. So students, with that we have answered the C part of the question. So we covered all the main components of the essential oils that we have seen in the previous question. So these are the main uses, or some uses, that you can actually come across when considering these main components of different essential oils. So now let's take a look at the second question. State three methods that can be used to extract essential oils. So we already know one method very well, that is steam distillation. So in this case, we are going to heat the plant material along with some water, and we are going to observe how the vapor that is produced in this process vape actually is going to contain both the water vapor and the essential oil, and in the steam distillation process we are going to condense this vapor and then obtain the mixture of water and the essential oil. And if these two essential oils and water doesn't mix, they will be separated out into two layers, and then we can easily extract this oil through removal of water using a separatory funnel. Or else you can actually, after even separating, if there is some amount of moisture remaining in the essential oil, you can again remove it by a drying method. And then there's another method known as solvent extraction. So in the solvent extraction process, students, we are going to take the plant material and try to extract the essential oil into an organic solvent. The essential oils that are found in plant materials are not very soluble in water. So we can't carry this process out with the use of water. We will use an organic solvent. So we will take the plant material and we will add it to a solvent where the essential oil will easily dissolve, right? So in the solvent that you use, the essential oil should be dissolving in that solvent very easily. If the essential oil is not very soluble in the solvent we are using, then the extraction process will not be a success. So students, you have to choose your solvent carefully. So you are using an organic solvent, and this organic solvent should be a solvent where in which the essential oil can easily dissolve in. And then after adding the plant material, we will shake this mixture, trying to extract most of the essential oils into the organic solvent. Now after extracting most of the essential oils into the organic solvent, we can evaporate the solvent until only the essential oil remains. Now students, to increase the efficiency of this extraction, or to increase the yield of this extraction, we can carry this extraction multiple times so that majority of the essential oil is removed from the plant material into the solvent, and then we remove the solvent by evaporating the solvent, allowing only the essential oil to remain. Right. So apart from that, we can have another method, especially when it comes to very volatile essential oils. We can use the pressing method. Pressing. So pressing method is used when we want to absorb the essential oil into another material. So essential oils sometimes get absorbed easily to things like wax. So what we do is we take two glass panels that are coated with wax, and then you can have the plant material that is going to contain the essential oil layered in between two glass panels that are coated with wax, and then we will press them. At this point, when you are pressing them, the essential oil will get absorbed onto the wax. After that, we will separate these two glass panels, and now the wax has the essential oil in it, and then we can extract the essential oil from the wax, so through a solvent extraction process. This process is a bit cumbersome, as well as it might introduce some wax in the final product, and it is not going to be a very efficient process. Not all of the essential oils are going to be extracted easily this way. So therefore, you do not necessarily see this final method being used in the industries a lot. However, especially when extracting cardamom oil, we use this method still. So cardamom oil is a good example of this particular method still being in use. And then we move on to the B part of second question. State the essential oil that is extracted from each of the plant materials listed below, along with the primary components found in each essential oil. So we have cinnamon, citronella and clove, right? So again, part of the plant used. Now, if you use the leaf, you are going to be extracting mainly an essential oil containing eugenol. Now, if you use cinnamon bark, you will be extracting cinnamon oil that contains mainly cinnamaldehyde, right? So you have to keep that in mind. So I have mentioned here leaf. So with the leaf, you get eugenol. Citronella. Citronella can be used, part of the plant. We can take Pangiri plant leaf, right? We can take the leaf of Pangiri plant, and again, the leaf is used for the extraction, and the main component of citronella oil is going to be geraniol, right? Citronellal is also there, but main component happens to be geraniol. And clove, when it comes to clove oil is mainly extracted from clove bud, right? And in that also the main component of clove oil is eugenol, right? So these are the primary components of these essential oils. So when it comes to cinnamon, if you write here and one additional detail, when it comes to cinnamon bark, you will have cinnamaldehyde. So cinnamon bark produces cinnamaldehyde, cinnamon leaf essential oil mainly contains eugenol, cinnamon bark essential oil mainly contains cinnamaldehyde. Briefly explain how essential oils can be extracted using methods other than steam distillation. Now, steam distillation method we did explain, and now we are expected to explain the other methods. So other methods also I gave you a brief overview, students. Here again, to give you a little bit of an overview, what we do is, when it comes to solvent extraction, essential oils are not very soluble in water. These are organic substances. They are not very soluble in water. So they will be soluble in organic solvents, right? You can choose a suitable organic solvent, and then this organic solvent that you choose should be able to dissolve or mix really well with essential oil. If the essential oil doesn't really dissolve in the organic solvent you use, the extraction is not a success. So you have to make sure the correct solvent is used, which can dissolve the essential oil really well in the organic solvent. So after choosing your organic solvent, we can add your plant material into that particular organic solvent. You can shake it, right? You can start by crushing the plant material if you like, and then you can add it to your organic solvent, and when you shake it, the essential oils will come out of the plant material and will dissolve in the organic solvent. And then organic solvent can be evaporated. Now we can first of all separate the plant material with the organic from the organic solvent now. So the organic solvent now contains the essential oil. So it's a mixture of essential oil and organic solvent, and then we can take it and evaporate the organic solvent. And as the organic solvent gets evaporated, the essential oil remains, right? So the essential oil will be the only remaining product here. The organic solvent will get evaporated. So students, that is how we use solvent extraction method to extract a essential oil. Now, when it comes to pressing, as I mentioned earlier, it is not something that we use quite often, but in this case we use plant materials that contain very volatile essential oils can be extracted this way. So you are going to take 2 glass panels that are coated with wax. Now when you place this wax coated glasses, and then you can layer your plant material with essential oils on top of the wax, and another plate or another glass panel with a wax coating can be placed on top of this plant material. and then you are going to apply pressure, a lot of pressure, until majority of the essential oil actually gets extracted into the wax. So as you press, the essential oils will come out of the plant material and will get absorbed into the wax, and then afterwards you can remove the two glass panels and the plant materials can physically be removed, right? So when you physically remove this plant material, the wax is now going to contain the essential oil, so we are physically removing the plant material. Now after that, we can carry out a solvent extraction, and even when you're carrying out the solvent extraction, you have to use organic solvent that will dissolve this essential oil really well in the organic solvent. And students, after that, you can use a rotary evaporator or a vacuum evaporator, and without subjecting the solvent along with essential oil to much heat, we can carry out a evaporation process to remove the organic layer or organic solvent. So in this process, we are not subjecting anything to a lot of heat because these essential oils that are extracted this way happens to be bit volatile. So then after the removal of the solvent, we are left with just the essential oil. So we first extracted the essential oil into wax through pressing, and then through the wax or whatever the essential oil that are extracted, the wax was extracted to the organic solvent through a solvent extraction process, and then the solvent was evaporated under milder conditions, allowing only the essential oil to remain, right? So this is how the pressing is done to extract essential oils. So if you explain those here, you can score the full marks that are allocated to this question. Then we move on to the third part, a bit of a calculation part. The essential oil named A is extracted from plant leaves through steam distillation method. So we are using a steam distillation method. The distillate contains both A and water. Derive an expression for the mass ratio between A and water, which is given as simple Ma, simple MH2O. At the temperature which the steam distillation takes place, the partial pressure of water is given as P0 H2O. The molar mass of A and H2O is given as Ma and MH2O respectively, capital Ma and capital M H2O. So now we have some information here, students. We have some information. Now they're asking, we want for the mass ratio between A and water in the distillate, right? Derive the expression for the mass ratio between A and water in the distillate. So that part has to be clear. We are talking about, we carry out the steam distillation process, and after that we obtain. Now imagine when you carry out the steam distillation process, students, we have the plant materials here, and then we have the condenser, right? We have the condenser here. We'll draw them a bit straight like so, right? We have the condenser, water in, water out, right? And then over here we have our distillate. Right, so over here we have our distillate. So in the distillate, we need to find out the mass of A to mass of H2O ratio. So because in the distillate we have a certain amount of water coming along over here, right? Over here we are going to have some water as well, right? So now how do we find it? The only information that we have been given is at the temperature at which, whatever the temperature here, the temperature at which this distillation takes place can easily be found out because when you have the thermometer included here, the temperature around this area can be read. So at that temperature, they say the saturated vapor pressure of water, right? Partial pressure of water, or in this case, the partial pressure of water vapor is basically going to be saturated vapor pressure of water. And saturated vapor pressure of water over here is pH2O, pH2O note. Hi, students. So now, if you observe what is going on here very closely, now if I magnify this and draw, imagine in this round bottom flask, which we usually use for processes like this, we have water vapor here. We have water vapor here, which I will indicate in these blue color dots, right? And their pressure is pH2O. And apart from that, we will have our essential oil also. I will indicate the essential oil in red. So essential oil vapor will also be there. So this red color dots indicates the essential oil vapor. So since there is essential oil vapor, there will be essential oil vapor pressure. Now essential oil was given as A. So therefore, we will say the partial pressure there is PA. Now, we know that the addition should be equal to atmospheric pressure, otherwise the boiling would not take place. And apart from that, students, we do know that when we are trying to carry out this steam distillation process, that vapor is going to go through this region, the same vapor. Now, this particular vapor will have a particular composition, right? There will be a certain percentage of A molecules here as well as water molecules here, right? There will be some composition. That same composition is going to be existing in this liquid form as well. Why? Because this vapor that I have indicated here is going to go through this Liebig condenser and get condensed into forming water. So if there is a particular number of mole ratio here, if the number of moles of essential oil is Na, and the number of moles of water is NH2O, the ratio between the number of moles that is found in the vapor phase as well as the liquid phase happens to be the same. Because what we do is we take this vapor, we send it through the Liebig condenser, and we turn it into a liquid. So it's the same vapor that turns into a liquid. So therefore, students, if you know the composition of this vapor, you know the composition of this mixture or liquid, right? So what you have to do is you have to write a small expression for this. You have to write a small expression for this. So you have to identify what the question asks. The question wants you to find out the ratio between the mass of water and mass of H2O, nothing more. So let's try to use that over here. So if you assume that each of these vapors actually behave as ideal gases, we can apply Dalton's partial pressure law. Actually in this case we can use the equation PV equals nRT, right? So we can write the partial pressure of the essential oil A is spread in this entire volume. Let's say this volume is constant. It is going to be the same volume where the water vapor is also going to be spread through. So volume is the same. Then we have N. So to provide this partial pressure of A, there should be, let's say, a specific number of moles of A, Na, and you have RT. So RT is the universal gas constant as well as temperature. So both the temperature and the universal gas constant for both of these vapors are the same, right? Then I'm going to, now let's say this is expression number 1, and we can do the same for water vapor as well. So if you write P H2O note multiplied by volume. Now volume, both of them are present in the same volume. So volume is also the same. And then number of moles of water can be denoted as NH2O. And then we have RT. RT in both cases are the same. So students, simply, if you write 1 divided by 2, right, expression 1 divided by 2, you are going to get to this answer, which is, you can see, you don't have to actually write it separately and then cancel off. You can see here itself that V cancels off. RT cancels off on both sides. So the final answer comes to P naught A divided by P naught H2O is equal to Na, number of moles of essential oil molecules, divided by number of moles of water vapor molecules, or water molecules in gaseous state. Right. So here essential oil molecules in gaseous state, here water molecules in gaseous state. Now this is the equation we are going to be left with. But students, we also know, now if you call this 3, we also know another very important equation. What is that? We also know that for any given equation, N equals mass divided by molar mass. Mass divided by molar mass. So for each of these, we can write this expression. So if you write Na this way, so we can write this expression 3 Pa divided by P naught H2O is equal to now Na. Na is equal to the mass of A in the sample divided by molar mass of A, right? That is for Na. Then we have NH2O. So the NH2O, now since it is in the denominator, you have to be careful. So NH2O, if you write it over here just to have better clarity, NH2O is equal to MH2O divided by simple MH2O divided by capital MH2O. Right? So since it is found as a part of the denominator, we can take the inverse of this 1 over NH2O, which is going to be M, capital MH2O divided by simple M H2O. So that is what should go over here, right? Simple M H2O divided by over in the denominator, and then in the numerator we have capital M H2O, right? So now at this point we have this equation. Now we need to get to this ratio, so we have to write or rearrange this equation, and when we do that we can just get the ratio between the mass of A and mass of B, or mass of H2O actually, is equal to, now that means we are going to only keep these two, is going to be equal to P naught A divided by P naught H2O, right? And here we have to send this over here. It will be capital MA, and this has to be sent over here. So at that point it will be capital M H2O, capital M H2O. So this is going to be the statement or the expression that you are going to obtain when you answer this question. So when you answer this question, this expression you get is this, because the ratio that they're asking is the ratio between simple or mass of A divided by mass of water, and when you do that we can obtain this expression. And you can see That is what they're asking here. Okay, and we obtain that answer by having an idea about what actually takes place when you're carrying out steam distillation. We are taking the vapor that is produced here and we are condensing it. So if you know the composition of the vapor, composition in the sense the number of moles of A as well as number of moles of water in the vapor, we can then directly identify the number of moles of water and number of moles of A found when they are condensed, because the same vapor is what is getting condensed here. So if you know the number of moles, we can find out the masses. So instead of just directly trying to find the number of moles, we wrote these two expressions, and using those expression and the expression about mass divided by molar mass, we obtain the ratio that was given in the question. So this is how we will carry out this calculation. So now we can move on to the next part. And now we are at the fourth question. It says naphthalene is extracted when steam distillation is carried out at 98 degrees Celsius temperature and 1.01 into 10 to the power 5 Newton per meter squared atmospheric pressure. The saturated vapor pressure of water at 98 degrees Celsius is 9.50 into 10 to the power 4 newtons per meter squared. Calculate the mass of the distillate that contains 10 grams of naphthalene after steam distillation process. So again, we are provided with a similar question to what we did in the previous part, but this is a different question, so we can't directly use that equation we derived. So we have to observe what is going on here. They have given us the saturated water vapor pressure at the temperature where the distillation is carried out. So they will try to develop an understanding here of what is going on. So we can do that through a small diagram. So we have our steam distillation setup, right? We have our steam distillation setup. Okay. Now over here we have our water along with H2O liquid here, along with let's say whatever extracted or whatever we extract naphthalene from, right? So in the vapor here, I will draw the vapor separately, and apart from that we have our condenser, right? This reads 98 degrees Celsius. And students, again, in this region you will have the vapor, water vapor. I will draw the water vapor like so. So in these blue dots, what I'm drawing is the water vapor. Now when you are drawing a small sketch, you don't have to draw it exactly the way that I draw here. This is mainly to explain what is happening in this process. So we can see this red color molecules that I've drawn here represent the naphthalene vapor. So in this case also, just to keep things simple, instead of naphthalene, the whole sentence, we'll call it A. It'll be easier for us to refer to. So basically this has partial pressure generated through the saturated vapor pressure of naphthalene here. Apart from naphthalene vapor, we have water vapor. We have water vapor here. P naught H2O. So now students, we know for this solution to come to a boil at 98 degrees Celsius, because water is there, water normally boils at 100. For this solution to come to a boil at 98 degrees Celsius, this happens because the saturated vapor pressure of the naphthalene along with saturated vapor pressure of water combined together is equal to the atmospheric pressure. So we'll first say it is referred to as the total pressure, and the total pressure should be equal to atmospheric pressure, P ATM, which is basically equal to, they have given the atmospheric pressure, which is 1.01 into 10 to the power 4, actually 10 to the power 5 Newton per meter square. They have given the pressure. So at 98 degrees Celsius, the saturated vapor pressure of water along with the saturated vapor pressure of naphthalene vapor is going to be equal to atmospheric pressure. So these two together is equal to atmospheric pressure, and the atmospheric pressure happens to be 1.01 into the power 5 Newton per meter square. So we can then simplify this and write P naught A plus P naught H2O is equal to 1.01 into 10 to the power 5 Nm minus 2, and apart from that, students, in this question the saturated vapor pressure of water at this temperature 98 degrees Celsius is given. Saturated vapor pressure of water at this temperature is given. What is the saturated vapor pressure of water at this particular temperature? 9.50. 9.50 into 10 to the power 4. So we can calculate the saturated vapor pressure of essential oil as well, or vapor pressure of essential oil can be calculated. So since we know the saturated vapor pressure of water, I'm going to now take it to the other side. So we can write here 1.01 multiplied by 10 to the power 5. Here we know this is going to be 9.50 into 10 to the power 4. Both of them are given in the same unit, Nm minus 2. Now you can't just subtract these two because the 10 to the power figures are different, so we have to bring them to the same value. In order to do that, we can jump this decimal place once to this side, so then this 10 to the power figure turns into 10 to the power 4. Let's do that. Let's do that and write for clarity. At the exam, if you are competent enough to do it on your own without having to rewrite it, you can go ahead and do that. I am explaining it here for better understanding, but necessarily this particular step, if you can eliminate it, it's fine. You can directly go to the answer because we have now substituted the values correctly. So then, students, after that, we can see the answer is going to be 0.6 into 10 to the power 4 Nm minus 2. 0.6 into 10 to the power 4 Nm minus 2. So this is going to be the vapor pressure of A found in this vapor mixture, right? So in this vapor mixture, we are going to have a vapor pressure of this much, 0.6 into 10 to the power 4 Nm minus 2. Now, at this point, we can use the knowledge we have to calculate, or we can actually use the same equation that we used earlier to get to the mass of naphthalene to mass of water ratio. Based on that, we can develop a good idea about the overall mass of the distillate. So if you do that here directly, if you rewrite this expression, because in the previous question it was a separate question, you can't bring that equation over here. So if you write an equation for the vapor of naphthalene, naphthalene was indicated by A, so vapor of naphthalene is spread throughout a volume of V is equal to Na RT. Na represents the number of moles of naphthalene. So this is our expression 1. And expression 2 would be saturated vapor pressure of water multiplied by volume. PV equals an RT equation is applied here. So volume is what? The same volume as the volume that naphthalene is spread through. So volume remains the same. Number of moles of water, of course, is different to the number of moles of A here. And RT, these are constants throughout this experiment. Temperature doesn't change. We keep it at 98 degrees Celsius, and R doesn't change. R is a constant. So at this point, I can rewrite 1 divided by 2, and we can see RT cancels off RT, V cancels off V here, and therefore we will get P naught A divided by pH2 equals Na divided by NH2O. Since we already know what we are going to be doing next, because of N equals M, because of this N equals mass divided by molar mass equation, we can rewrite this equation in the following way. Mass of A divided by molar mass of A, and then we have over here NH2O, so then it will be mass of H2O in the sample divided by molar mass. So when divided by molar mass means in this case it will be brought up into the numerator. It will be molar mass of H2O here. So students, if you get mass of A to mass of water ratio, we get an idea in a given distillate what is the mass of water as well as what is the mass of the naphthalene produced or naphthalene extracted, not produced, naphthalene extracted. So we can do that here. Ma MH2O is equal to P naught A divided by P naught H2O, and apart from that, students, we have these masses. Ma goes up here, and you have MH2O coming down here, MH2O. So this is the expression. We developed this expression earlier as well, but only difference in this particular question is we are going to apply this with correct values and find the ratio, this correct ratio. So students, when you apply these values, we know P naught A at 98 degrees Celsius happens to be 0.6 into 10 to the power 4 Nm minus 2. and P naught H2O happens to be 9.5 into 10 to the power 4, and the unit is the same. Molar mass of A should be found out, and molar mass of water we already know, so we will substitute it here. It's going to be 18 grams per mole. That is the molar mass of water. And molar mass of naphthalene should be found out. So molar mass of naphthalene. So since naphthalene formula was given in the question, so although we haven't written it down here, there was a naphthalene formula given, C10H8. The naphthalene formula is C10H8. So that formula is given in the question, otherwise you are not required to necessarily remember the entire formula of the naphthalene molecule. So here, since you know the formula of naphthalene, you can calculate the molar mass of naphthalene. So C10H8. So now students, each carbon is going to be having a value of 12, or atomic mass is going to be 12. So there are 10 of them. Therefore, this is 120. 8 hydrogens are going to bring in a value of 8 here. One hydrogen is 1, so there are 8 of them, 8. So the molar mass is 128. 128. So over here we can write that the molar mass is 128 grams per mole. Grams per mole. We can see grams per mole cancel off, and here Nm minus 2, Nm minus 2 cancels off. Apart from that, students, you can also observe that this cancels off. 10 to the power 10 to the power 4 on each side, they cancel off. So right now we are left with only these terms. So if you proceed to solve them, you would see this cancels off, leaving behind only a 3 here, right? So we have 0.1 multiplied by 128, and in the numerator, in the denominator, we have 9.5 multiplied by 3, right? So if you go ahead and write them down, it's going to be 12.8 divided by 28.5. 12.8 divided by 28.5. 3 multiplied by 5 is 15, and 27 plus 1 is 28. So this is the ratio that you get, right? So this is the, you can further solve this, right? Mass of A divided by mass of water. Mass of A divided by mass of H2O. So we can work with this ratio as well, or else if you really want, you can further solve it, or you can keep the solving to last. So now in this question they say in the distillate, they say in this distillate they say there is 10 grams of naphthalene. Naphthalene, we assigned a letter A for naphthalene here. So we have 10 grams of naphthalene, or A. So if you know the mass of naphthalene and if you know the ratio, can you not find out the mass of water that is found here? We can. So let's go ahead and do that. If the mass of naphthalene is known, which happens to be 10 grams, we can isolate MH2O. If you know the mass of naphthalene, then using this equation, we can find out the mass of water in that particular distillate. In a specific distillate, there was 10 grams, but the ratio is going to be always this. We can further solve it, but since they are not asking for the ratio in this question and they are asking for the mass of the entire distillate, we don't have to proceed to solving this any further. We can keep it as it is and proceed to finalizing the answer. So since this is 10, we can find out M. So first we'll MH2O. So first let's isolate MH2O and rearrange this equation. So we can write MH2O is equal to, so we take M over here, H2O over here. So that means we have MA multiplied by 28.5 divided by 12.8. So we know MA is 10. So directly we can just write that here, 10 grams. Now this is coming in grams. So the final answer here will also be obtained in grams. So then this becomes 285 divided by 12.8. The answer comes in grams. So students, when you carry out this calculation, you will get the answer. We get to 22.26, right? This many grams will be obtained. We can round it off to 22.3 grams. We have 22.3 grams obtained here. So when you solve this, we get the mass of just the H2O. So the question is, when you have a distillate that contains this many grams of A, what will be the mass of just water? So mass of just water we just found out, in that distillate it should be 22.3 grams. So in this distillate the mass of water is 22.3 grams. So this is the mass of just naphthalene. The question that is asked is the mass of the entire distillate. So the mass of the entire distillate is going to be given here. We can write MT. Mass of the entire distillate is equal to mass of A plus mass of H2O. Now A denotes naphthalene. So mass of A is 10 grams and mass of water happens to be 22.3 grams. So when you add them together, the answer that we obtain is 32.3 grams. 32.3 grams. So this is the final answer to the question that is asked. So what we did here is, students, now we will first copy down this answer over here, the answer to this question. Calculate the mass of the distillate. So the mass of the entire distillate, we can write mass of the distillate is equal to 32.3 grams. Right, so how did we do it? Again, we looked at the vapor that is found here in the steam distillation process. We knew the vapor consists of saturated water vapor and the essential oil, or in this case naphthalene, that is present, right? So naphthalene that was present here was initially named as A for convenience when working with these different types of symbols here. And then after doing so, we can actually find out that the vapor pressure of water along with the vapor pressure of naphthalene is equal to the atmospheric pressure, and with that we found out the vapor pressure of just naphthalene, right, which happens to be 0.6 into 10 to the power 4. And afterwards, students, we got to the mass of naphthalene to mass of water ratio in the final distillate. And to obtain that, we took a look at PV equals nRT equations for each individual gas that is present in this vapor. And then through dividing these two equations, we got to the molar ratio in the vapor. But if you know the molar ratio, you can get to their mass ratio. So since we know the molar ratio, we isolated its mass ratio using the information we have at hand, and then now we knew the partial pressure of naphthalene as well as the saturated vapor pressure of water, and we know the molar mass of naphthalene, we know the molar mass of water. So based on these findings, we were able to finally calculate the mass of water in a distillate that contains 10 grams of A. So this is a distillate, right? So in this distillate, they say there are 10 grams of A. So if you have 10 grams of A, using the relationship we have obtained here, we can calculate the mass of water that is found in the same distillate. So with that, we were able to find the mass of just the two components found in the distillate. So if the distillate contains only these two components, then we know the total mass of the distillate is going to be addition of these two masses. And that is how we obtained this answer. So students, this is how you can attempt a question that is revolving around experiment number 42, or that is revolving around steam distillation. So with this, we hope you have developed a good enough understanding of how to attempt theory-based questions associated to these extraction processes, as well as calculation-based questions. So with that, we can actually conclude today's experiment, and I will be meeting you again with the next experiment.